Hi, this is Dr. Collins Jones. Welcome to segment 4 of my video series on manual cell counting and cell density calculations. In this segment, we will cover how to do the actual calculations of percent viability and cell density. We will also briefly review scientific notation and how to determine and calculate dilution factors. So let's get started with some basic definitions of percent viability and the cell density. That is, the equations that we use to do these calculations. So again, percent viability is the number of live cells divided by the number of total cells times 100%, right? And so recall, the number of total cells is the number of live cells plus the number of dead cells. This is the data we get when we do the actual counts using the hemocytometer and the microscope. Remember, the live cells are the cells that have a clear cell membrane outline but are transparent or translucent, and the number of dead cells are the ones that are stained with our vital dye, and in our case we use trypan blue as the vital dye. The cell density, or the number of cells per 1 milliliter, is the number of total live cells divided by the number of squares counted, which if you're doing this the way we recommend, that would be 10 squares, but suppose you count 5 squares, then it would be per 5 squares, times the dilution factor, so DF is dilution factor, times a correction factor of 10 to the power of 4. Okay, and so the 10 to the power of 4 is really 10 to the power of 4 milliliters to the minus 1, or that's 10 to the 4th over milliliters. This is the correction factor for the volume of cells being counted in the hemocytometer, providing that you load the hemocytometer correctly. Okay? The dilution factor is then used to correct for the increased volume that we get when we dilute a cell suspension. And if we increase the volume the cells are suspended in, that decreases the concentration. So we need to get back to the original concentration before dilution. So to make that correction, the larger volume, V2, is always in the numerator, and the smaller volume is always in the denominator. So dilution factors are the final volume over the initial volume, or V2 over V1. There may be more than one dilution that must be corrected for, and we have to include all of them in the calculation by multiplying each dilution factor times the other dilution factors. We're going to illustrate this with some examples specifically dedicated to calculating dilution factors before we actually get into a calculation of cell density and percent viability. In our labs, the dilution factor should always be at least 2 over 1 because we mix equal volumes of trypan blue with equal volumes of cells. That is, we mix 50 microliters of trypan blue with 50 microliters of cell suspension, and so the final volume would be 50 plus 50, right, which is 100. That's the combined trypan blue and cell suspension, divided by 50 microliters, which is the initial volume, just the cell suspension. So 100 divided by 50 is 2 over 1. So just to get our brains in gear that, when we talk about cell densities or cell concentrations, these can be fairly large numbers. So cell concentrations are usually recorded, or if you will, reported using scientific notation. So an example here could be anchorage-dependent cells, and a typical number for anchorage-dependent cells may be 5.49 times 10 to the 5th cells per centimeter squared. And sometimes when you have to do calculations and conversions, it's helpful to remember that if we see this 5.49 times 10 to the 5th cells per centimeter squared, that's really 5.49 times 10 to the 5th cells over 1 centimeter squared. Okay? And so you're thinking about this. This means for every 1 centimeter squared of surface area on the T-flask or a cell factory like a Nunca or Hyperstack, there are 549,000 cells per 1 square centimeter, right? This is a surface area, where the surface area is length times width. For suspension cells, a typical number may be something like 3.75 times 10 to the 6th cells per mil. And again, if it helps during conversions, it's really 3.75 times 10 to the 6th cells per 1 mil. And here we are actually in a volume, so this means for every 1 mil of media, there are 3,750,000 cells. And if you get a really high density, this can go as high as 10 to the 7th or 10 to the 8th in some cases. So again, here we're dealing with a volume, right? Length times width times height. And so within this 1 cubic centimeter or 1 mil volume, there are 3.75 times 10 to the 6th cells. All right, a quick review of scientific notation. If you are working in cell culture, you should have been at the point where you know scientific notation, but we just quickly want to review that 10 to the 0 power is equal to 1, 10 to the 1 is 10, 10 to the 2 is 10 times 10 to the 10th, or 10 to the 1 times 10 to the 1, which is 10 to the 1 plus 1, or 10 squared, which is 100. 10 cubed is 10 times 10 times 10, or 10 to the 1 times 10 to the 1 times 10 to the 1, or 10 to the 1 plus 1 plus 1, or 10 to the third, which is 1,000. And hopefully I don't need to go through the rest of these, but you get the idea. If you are going to do calculations in cell culture, you should be comfortable with scientific notation. To enter any of these powers of 10 in your calculator, you're going to use the EE, which is enter exponent key, or the EXP, the exponent key. So when you press an EE button on a calculator or the EXP button, that's like putting in times 10 to, and then you see the question mark in red, times 10 to whatever power. So you don't need to say, for example, 5.75 and then go times 10 and then press EE and then go to the third, because that's really going to be 10 to the fourth, right? So for example here, if you were going to enter 3.75 times 10 to the 6th, you would first key in 3.75 and then press the EE or the EXP key and then just enter in 6, because the EE or EXP is the times 10 to whatever. Right? So just be aware of that. If you're always off by a power of 10, that is, you get 10 to the 7th instead of 10 to the 6th, it's probably because you're entering it into your calculator incorrectly. Okay. All right. I know that was quick, but hopefully you guys are all comfortable with this. I bet some of you even skipped this slide and fast forwarded, and that's fine. These next two slides, though, you should take a minute with, because invariably I find that my students struggle with dilution factors, even the ones who come to us with 4-year degrees, master's degrees, and PhDs. So first thing is, why do we need to dilute when we do cell counting, right? Because we need to have the counting range of 20 to 50 cells per square. Less than 20, it's not accurate, and more than 50, we generally make the assumption that you can't accurately count all of the cells in the square. There are too many cells to differentiate between individual cells, so you may skip counting a cell, or you may count the same cell twice. So we need to dilute the cell suspension to get the range of cells into the 20 to 50 cells per square. But if we do the dilution, we need to correct for the dilution, right? So the general formula that we use to calculate dilutions, you guys are all familiar with this. It's C1V1 equals C2V2, right? If you haven't seen this before, but you've had chemistry, you saw this as M1V1 equals M2V2. That's for a specific case of molarity. This is the general case for any concentration. So again, C1 is the initial concentration, the highest concentration. This would be the undiluted cell suspension. V1 is the initial volume, that is the smallest volume. This would be the volume of suspended cells that you need to dilute. C2 is the final concentration, right? It'll be the lower value. That'll be the concentration of cells after the dilution, and V2 will be the final volume. That's the volume that the cells end up with that you use to count. So the important thing to recall here is that C1 and C2 must have the exact same units, and V1 and V2 must have the exact same units. And when we're doing these kinds of calculations for determining the cell concentration, is really difficult to not have C1 and C2 in the same units. They're either going to be cells per mil or cells per centimeter squared, and the same for V1 and V2. V1 and V2 are almost always going to be in values of microliters, all right? But if you're doing other kinds of calculations, these units may differ, and you need to just make sure that C1 and C2 are exactly the same, and V1 and V2 are exactly the same. Now, the other thing, especially when you're working dilution problems, that is written problems, but even if you think about it, as you do the dilution, you're using the word to or you're using the word with. And it's important to pay attention to this distinction, right? This is a way to determine if the second volume is the total volume, that is, we say diluted to, that means that's the final volume. Or if the initial volume and the volume that we're adding need to be added together. That is, if we're combining two volumes and we say we are diluting 75 microliters with 200 microliters, and now we need to do 75 plus 200. That's what we're going to illustrate on the next slide and work through the calculation. Okay, so if you struggle with dilutions, spend a few minutes on this next slide. So here are some examples of the to versus with. All right. Determine the dilution factor if 75 microliters of cells are diluted to a final volume of 375 microliters with 1x PBS. And if you're in my classes, you need to show all work so that we can go back and, one, give you partial credit if you got something wrong, but two, if you're working at a company or in a lab and somebody audits your work, they can easily follow your calculations. Now, I've used some sort of non-traditional numbers here just to illustrate to you that it doesn't matter what two numbers you have in your dilution scheme. As long as you know the initial volumes and the final volumes, you can always calculate a dilution factor. It might not be a nice, neat number, but if you made a mistake in adding the volumes, as long as you keep track of them, you can always correct for the dilution, right? So in this case, we see that we are diluting to a final volume of 375 microliters, right? So the initial volume is going to be, that is V1 is going to be 75 microliters. And so since we said diluted to, the final volume is 375 microliters, our dilution factor is always V2 over V1. So that's 375 divided by 75. And you can simplify this, but I usually just leave it like this because the more extra math steps you take to simplify things, sometimes the more likely you are to make a mistake. And that might even include mistakes in round off errors. Remember, in the rules of significant figures, we don't round off until the final calculation. Okay, so 2 tells us that the second volume is the final volume, and the first volume we are given is the initial volume, right? So final volume is V2, initial volume is V1, and the big number always goes on top so that we can get back to the original concentration. All right, next, determine the dilution factor if 75 microliters of cells are diluted with, right? So here we have with 375 microliters of 1x PBS. All right, so now the with means we are adding the two volumes. So V2 is 75 microliters, the initial volume, plus 375 microliters, the volume that we added the 75 microliters to. So our final volume is 450 microliters. But again, the initial volume is 75 microliters. So our dilution factor is V2 over V1, but now it's going to be 450 over 75. Now the next one is when we had to do multiple dilutions. So it says determine the dilution factors, plural, if 25 microliters of cells are diluted to 225 microliters with 1x PBS, and subsequently 50 microliters of this first dilution is removed and further diluted to 800 microliters with 1x PBS. In an upcoming segment, I will actually do this again, and I will illustrate what it would look like in the lab, sort of using my little animations and cartoons. But right now, we just sort of want you to get the idea of what it's like to work with two dilutions simultaneously. Okay, so two dilutions were completed, and this means we must determine two dilution factors that need to be combined. So dilution factor 1, right, we have 25 microliters diluted to 225 microliters, so that means that 225 is the final volume. So this will be 225 over 25. That's our first dilution factor. The second dilution factor says that 50 microliters of the first solution is removed and further diluted. Again, we have two 800 microliters, so since we diluted this two 800 microliters, it's going to be 800 divided by 50, and that means that we had to take this 50 microliters and mix it with 700, and notice the word mix 50 microliters with 750 microliters to get our 800 microliter final volume. We just didn't say that in the problem. We simply said it was diluted to 800 microliters. But 800 minus 50 tells us what volume of diluent, that is the PBS, the solution we're using to dilute with, was required. Okay, so now the total dilution is going to be dilution factor 1 times dilution factor 2, or 225 over 25 times 800 over 50. And again, if you prefer, you can simplify this and divide the denominator into the numerator to get simpler numbers. All right, but I generally do the calculations just like this, especially if you have this in an Excel spreadsheet or some other auto calculator. All right, another one. Determine the dilution factors if 100 microliters of cells are diluted with 400 microliters of PBS, and subsequently 50 microliters of this first solution is removed and further diluted to 150 microliters with PBS. So in the first one, we are combining 100 microliters with 400 microliters. So 100 plus 400 gives us a final volume of 500. So V2 is 500, 100 plus 400. Okay, and V1 is what we started with, 100 microliters. So that's our first dilution factor, 500 over 100. The second dilution factor, it says we are diluted to 150. So V2 is 150, and again, V1 is 50 microliters. So that's our second dilution factor. To back calculate and account for the overall dilutions to get to the original concentrations, we need to combine the two dilution factors. That is, multiply dilution factor 1 by dilution factor 2. So this is 500 over 100 times 150 over 50. Okay, and then finally, I want you guys to read this, pause it, see if you can work it out on your own and just come up with the dilution factors. I'll count to 3 and then start recording again and see if you can come up with the right answer. Okay, back. Determine the dilution factors if 200 microliters of cells are diluted with 400 microliters of cells using 1x PBS, and subsequently 25 microliters of this first dilution is removed and further diluted with 175 microliters of 1x PBS. Okay, and again, show all work. So here, both times we have with, so that means that each dilution, the volumes need to be added together. Okay, so dilution factor 1 is going to be 200 plus 400 for our final volume, right? 200 microliters of cells are diluted with 400 microliters of PBS. So the final volume is 600. Again, 200 plus 400. And the initial volume was 200 microliters. In dilution factor 2, we also used with. So we have 25 microliters combining with 175 microliters of PBS. So 25 plus 175 is 200. And our starting volume was 25. So V2 is 200. Again, 25 plus 175 divided by the initial volume, 25 microliters. So the total dilution is going to be dilution factor 1 times dilution factor 2, or 600 over 200 times 200 over 25. So that was a quick review of dilution factors. Hopefully you found this useful, or maybe you already knew how to do this. But in either case, we can now proceed to do calculations of actual percent viabilities and cell densities. So let's really start with just getting the raw data. So here again, I've made a cartoon picture, if you will, of a hemocytometer, of one of the counting chambers in the hemocytometer with all of the squares occupied by cells. So the problem states determine the percent viability and the cell densities, that is the number of cells per mil for a cell sample that was diluted 1 to 2 with trypan blue, and count it. All right, to save time and space for this problem, we're only going to use squares 1 and 2 for your calculation. Ideally, we'd use at least 5 squares, but in order to get through these examples, I'm going to limit us to 1 or 2 squares. Okay, so square 1, first thing we're going to do is count the cells in squares 1 and 2. So here's square 1. Remember that we have to be mindful of what the actual square is, right? The interior that we're going to count. We're then going to do the count, right, in a regular counting pattern. And if we do that count, and you guys should have done this yourself, right? We find that we have 21 total cells, 3 of which are stained blue, so those cells are considered to be dead. So 21 minus 3 will give us 19 live cells. And then we do the same thing in square 2. Again, keeping in mind that we're only counting the interior, and this is outside of the counted square. So again, we use our counting pattern, right? And in square 2, we see that we have 24 total cells, and again, 3 dead. Okay, so we have a total of 45 cells, right? 21 plus 24 is 45 cells. And we have a total of 6 dead cells, right? 3 plus 3 is 6. And then 45 minus 6 gives us 39 live cells. So now we have all the raw data we need to calculate the cell viability, the percent viability, and the concentration. So to calculate the percent viability, this is just a simple plug and chug, right? It's the number of live cells divided by the number of total cells times 100%. So it's 39 live divided by 49 total times 100%. And notice in calculating the percent viability, the dilution factors don't matter because it'll be the same ratio of live to total. All right. So the percent viability here is 86.7%. And when we do our calculations with the cells, we generally stick with 3 significant figures here. Okay. So that's just by universal agreement. So our percent viability is 86.7. And if you want to comment on that, 86.7%, it's not great, but it's not bad. But often in manufacturing situations, we are shooting for 95% viability or above. If we're not above 95%, if we're not at or above 95%, oftentimes that means it's out of specification, and we need to do a root cause analysis to figure out why the cells are not growing to the percent viability we want. But here, again, very simple. Live cells divided by total cells, 39, which we counted, divided by 45, which we also counted, times 100%, gives us 86.7% viability. To calculate the cell density, it's really no more difficult. We look at our equation to calculate the cell density of the cells per mil. It's the total live cells divided by the total squares counted times the dilution factor, V2 over V1, times the hemocytometer correction factor, 10 to the power of 4. So we just plug this in, right? It's 39 live cells from here. We counted 2 squares, right? Square 1 and square 2, so it's 39 divided by 2 times our 1 to 2 dilution factor, right? So this is going to be 2 over 1 in order to account for the fact that we made the number of cells half, times, right, times our hemocytometer correction factor. So we multiply all this out, and this turns out to be 3.90 times 10 to the 5th cells per mil, and again, 3 significant figures. Okay, so not that hard. Okay, we're going to end this segment with just mentioning sources of error in cell counting. So these are mistakes that can be made in cell counting, and that you should be aware of these and do your best to avoid them. But if you get incorrect counts, if the counts don't look right, these could be some of the reasons for it. We've said this multiple times. The microscope is not focused. The microscope is not clean, right? If it has dust on it, that may be mistaken, especially by a beginner, as a dead cell. Mixing issues. We haven't resuspended the cells. So if the cells have settled and you take either from the middle or from the bottom, it's not going to be representative of what was actually in the flask. If you go to respend the cells and you pipette up and down vigorously, if you triturate vigorously, that is, you draw the cells up and down the pipette, that can create shear, and shear will stretch the cells out and they will burst open and die. Same thing, we never vortex mix in cell culture, right? That vortex creates lots of shear as the cells are caught in the vortex around the edges of the microcentrifuge tube. And then, of course, we can have clumps of cells, which we talked about earlier. The way to account for clumps of cells should be indicated in the counting protocol, but they are difficult to correct for. Then we could have too high a cell concentration. Then you have to do your dilution, right? And again, we recommend using PBS or HBSS. If the cell concentration is too low, you may actually have to concentrate the cells by centrifugation and resuspending them in a smaller volume. And again, remember, there should be 20 to 50 cells per square. There should be 20 to 50 cells per 1 square. Then if we deal with trypan blue, incubation times, if it's too short, right, non-viable cells, that is dead cells, all of the non-viable cells may not take up the dye if you don't incubate for at least 2 minutes. But if you wait too long, like 10, 15 minutes, then viable cells, that is live cells, may also take up the dye. As we stated earlier, trypan blue has some solubility issues. There may be crystals in the dye solution that may be miscounted as dead cells. So when possible, we recommend filtering your trypan blue solution right before counting, but that's not always practical. But I get to tell you the best practices, and it's up to you to decide if you want to use those or not. Then hemocytometer issues, if it's not cleaned, we may have debris from previous counts, or there may even be some stained cells on there that will make your count incorrect. If it's not filled correctly, we don't get the right volume, so again, the concentration will be incorrect. And if somebody used an abrasive to clean the hemocytometer and it's scratched, those scratches actually change the volume of the hemocytometer, and they may change how the cells distribute. Then there are pipetting errors, again, right? Incorrect volume setting when you do your dilutions. Bubbles are troubles in the hemocytometer, and we talked about overloading the hemocytometer. And then if you don't use the correct buffer to dilute, you can actually kill your cells. So again, we recommend 1x PBS or 1x HBSS. Some people use media. I don't really recommend that. One, because it's expensive. Two, you risk contaminating the media. Three, there may be a component in the media that changes the way things stain. And then some people, again, dilute exclusively with trypan blue. If that's what the SOP says, that's what you should do. But again, that's not what we recommend. We recommend using a balanced salt solution like PBS or HBSS. These are really cheap, but they maintain the correct osmotic strength so that the cells don't die. And that is the end of segment 4.